So, course-wise, housekeeping. I feel like last time I went a little bit fast. So I think what I will do this time is recap from the beginning of amino acid metabolism. I have like a very short PowerPoint on like linking different pathways in metabolism that I've now already uploaded to your drive. We'll finish that today. There probably, my guess is there should be about like 10 to 15 minutes at the end of class today to ask like potential questions you have broadly. And then on Wednesday, I will use that as a class extra help set. I know that there's like a lot of material on this test, right? With every new pathway, it seems like there's another overlap between like a different pathway, and in that way the material can compound on itself. So I think rather than cramming in another lecture, we can just dedicate more time locking down the pathways that we have already covered. So trying to err on the side of quality versus quantity here. Any questions housekeeping-wise? Broadly? Nothing? Okay. So just to reiterate, we will finish amino acid oxidation today. We will talk about linking different metabolites, at least in just a couple of different pathways, and then on Wednesday, again, we will have a live extra help session with a QR code. So now, let's do a quick review. So kind of the back half of this slide is just talking about, or back half of this PowerPoint is just broadly talking about the chemical logic of nitrogen metabolism, right? Essentially establishing when would an organism actually need to break down amino acids, right? And different organisms will require their energy needs to be derived from amino acids according to the organism's diet, essentially, right? And on a prokaryotic level, it can vary pretty dramatically. But focusing on human cell metabolism here, or let's just start with terrestrial vertebrates, right? So you have carnivores, which might be predominantly eating meats, and therefore you would expect that they would have upregulated nitrogen metabolism, right? As opposed to herbivores, you would expect would not have such a high nitrogen metabolism, right? Obviously we're omnivores, so we have to balance this larger equation. So there are essentially two different ways that we can use or derive our amino acids, right? Either they can be consumed in your diet, or alternatively, they can be featured in regular protein turnover. So even if you were to not consume any proteins at all, there would still need to be this continuous process of protein breakdown and translation that would need to occur in cell maintenance by default. So again, you'll need to have protein turnover regardless of your anabolic needs or catabolic needs. So we did not talk about the peptidases specifically. You do not have to memorize these, but bigger picture here is that you can have, or you have, a series of specialized enzymes that are aimed at breaking down your larger polypeptide structures, right? Given that polypeptide structures can be so varied and feature different amino acids, you have peptidases that are specific for certain kinds of amino acids, right? And those kinds of amino acids can either be based off of the trait of the amino acid itself, or alternatively, where that amino acid is actually located on your polypeptide chain. Is it at the N terminus? Is it at the C terminus? Is it an aromatic amino acid or not, right? So you can have different peptidases that are specific to either the position of the amino acid or the kind of amino acid you are looking at. But you do not at all have to memorize the peptidase, right? And I think thinking on an organismal level, right, we have our human stomach, right? So our breakdown of proteins can happen in the stomach or in the small intestine. Our stomach will have peptidases that are less specific for specific kinds of amino acids and just kind of broadly break down these larger proteins, versus your small intestine will contain a lot more proteases that are specific towards breaking down those individual amino acids. Again, as far as I'm concerned, this is not super high yield for me. I much prefer metabolism on like the cellular level, but this background information is there if you might need it, or if it is helpful for you if you're wondering, where are these proteins coming from, or if you're a pre-med or pre-PA person, just kind of thinking about how it is relevant to your specific area. And kind of interestingly enough, we see like a theme that continues both on an organismal level and even like a subcellular level, this idea of creating excess folds to increase surface area to facilitate reactions that would happen along that line, right? So on an organismal level, we see that both in the folds of the stomach and the small intestine, but on a cellular level, we see that, for example, with the cristae in your mitochondria. And even within the context of that newer research paper, kind of talking about how there are mitochondria that lack these cristae that are more responsible for these anabolic So, as I've kind of mentioned before, when we thought about respiration, glucose metabolism, whether that's an TCA cycle or glycolysis, I oftentimes ask you guys to follow the carbons, or another way of saying that is like follow the carbons or follow the reduction sources, right? Or the sources of reduction there. Because the key side product that you're generating from these oxidative reactions are your NADHs and your ATPs, right? Alternatively, of course, you can use these carbon skeletons for a variety of different things and anabolic processes, but we did not delve into those excessively. But we did talk about gluconeogenesis, we did talk about fatty acid synthesis in the asynchronous lecture. But linking these two different processes, the citric acid cycle and the urea cycle, you have the aspartate arginosuccinate shunt of the citric acid cycle, and we will talk more about that a little bit later. So again, while I don't intend for the third exam to be cumulative, like I'm not going to drill you guys on the regulators of citrate synthase. Don't completely dump your brain of your understanding of the TCA cycle, because you will see a lot of those metabolites have overlaps and shunts into your urea cycle, into nuclear metabolism, which we will cover a little bit later today. So, this is just outlining again what is the general chemical logic of the movement of these nitrogen groups. And this can happen in the liver of different organisms and is not specific to humans by any means, right? So again, you have three different sources of your proteins here, right? One, these amino acids can be directly, locally within the liver, within the hepatocytes, right? We're thinking that this is occurring within a hepatocyte, right? That is the big asterisk here, that the urea cycle is occurring within a hepatocyte. But broadly, in livers of different organisms, whether that be a fish, a human, or a bird, you would expect a series of oxidative transaminations to be occurring. And again, you can have local amino acids. Those local amino acids can either be from normal protein turnover or from digestion and transport to the particular liver cell. And then you also have two other pathways via alanine, right, which we talked about as a kind of like 2-in-1, where you can get rid of nitrogen and excess pyruvate with one molecule. Or alternatively, you can have glutamine, which stores two levels of nitrogen, right, or two nitrogen molecules that can then be shunted into the urea cycle. So again, on a wide organism level, you can have amino acids from protein turnover or consumption. If we're thinking within the context of the liver, you have three different entry points, which is normal protein turnover or consumption, or the transport from other tissues, either in the form of alanine or glutamine. Any questions about that so far? Okay. So this is broadly just talking about other kinds of cycles or processes that other organisms might undergo. Again, just talking about 3 different molecules here. You have your straight up ammonia release that occurs via an anion or a cation. You have the release of urea, which we notice again is 2 nitrogens on either side of a bicarbonate. Or alternatively, you have uric acid, which contains 4 nitrogens and is a much larger molecule. And kind of thinking broadly, the reason why we even have the urea cycle is because humans are not optimized to dispose of this nitrogen in any other way, right? Fish have gills and other such ways of interacting with the environment to actually just release this ammonia directly. Humans do not have this luxury. So we have to actually undergo the urea cycle because we don't want free-floating ammonia molecules going throughout our bloodstream, as they are highly, highly reactive. So, this again is just like talking about general chemical logic here. So this is just mentioning that you are going to have the movement of amine groups that happens via enzymes, right? Or otherwise known as enzymatic transamination. It will use a pyrophosphate, nothing new there. Alpha ketoglutarate will be used to accept the nitrogenous groups. Glutamine will be your most common storer of nitrogen, right? So if you're thinking tissues broadly, glutamine is probably going to be how you're going to be transporting those nitrogens to the liver. Alternatively, alanine will be used from muscle cells specifically, right, or skeletal muscle cells specifically. But given that glutamine has this vital function across all of your cells in your body, it will be present in higher levels. Alanine will be also present in some degree to higher levels, but not nearly to the same extent as glutamine, right? And this is just generally outlining that same process, right? These transamination reactions. And here we're just seeing our first iteration of this transamination reaction, where you have the movement of a nitrate or nitrogenous group onto the alpha ketoglutarate molecule, now generating the amino acid L-glutamate. And you're left with an alpha keto acid within your cell, right? So again, just outlining the general chemical logic here. Now, that glutamate can then be oxidized, right? It can then be oxidized. This is an oxidative deamination reaction. That oxidation can use either NAD or NADP as that co-substrate. Now, keep in mind, this only really talks about glutamine, right? We can facilitate a similar process with glutamine, right? Except when you deaminate glutamine, you're removing that nitrogen from the variable R side chain, and you are left with a molecule of glutamate, right? So you can do 2 levels of deamination with glutamine versus only 1, as dictated on this slide, with your glutamate. And you're regenerating your alpha ketoglutarate molecule. Yeah?
Why don't we just focus on those 2 amino acids instead of the rest of the 20, if all of them have that amino on them?
Like, why don't the other amino acids, why aren't they used for? Well, I'm not familiar with every single amino acid synthesis pathway there is under the sun, but different pathways have other functions. Say, for example, in that recent research paper that I shortly presented, the synthesis of proline can be used in actually now deciding mitochondrial fate, right? So we have, 1, we're not fully aware of the other functions that all these amino acids, or the synthesis of these amino acids might have. And 2, the actual backbone of these structures align very well and easily with TCA cycle intermediates that we can then use again. So first is, I don't know. It's very possible that there are other pathways, but the more immediate answer is we can see right here that it more directly interacts with the TCA cycle and has more overlap.
Okay.
Yeah.
Do you think it also has to do with glutamate being polar in
honestly, blood biophysics is like way above my pay grade. It very well could be the answer, but like blood biophysics is not something I will pretend to know. It's like one of the big, like, it's very difficult to model and to understand, so I don't even know much about it. But that very well could be possible. Yeah.
I guess the side question I'll ask is, if the only thing we're focusing on with glutamate is that amino group, could theoretically all those other amino acids, technically speaking, go through the same process?
Absolutely, other amino acids can go through oxidative deamination for sure. And we actually talk about that like later on. Like there's other feeder pathways into, but ultimately we use glutamate as that origin for nitrogen in the urea cycle, and it's advantageous biologically as we don't have to use different enzymes within the mitochondrial matrix. We can just kind of converge on glutamate, and we don't have to worry about all the different transporters.
Which one's the most abundant in the use of the cycle, glutamate or glutamine?
Glutamate, without a doubt, is most important to the urea cycle
But
because
it's
it's
more beneficial to do glutamine.
glutamine is more efficient at storing nitrogen simply because you can store 2 nitrogens in glutamine versus glutamate only has 1. So rather than making 2 molecules of glutamate, rather make 1 molecule of glutamine, right? But you use glutamine in the urea cycle, or sorry, glutamate in the urea cycle because you can easily regenerate alpha ketoglutarate. And I guess it'll make more sense when we go to that slide, but it'll make more sense shortly. Yeah.
I'm confused. So when are we going from glutamate to alpha ketoglutarate? Like when are we utilizing this process if we're going the other way for the introduction into the urea cycle?
It might be a little bit better if we just jump to that slide, but it's over here essentially. So we will regenerate alpha ketoglutarate from glutamate within the hepatocyte level. And here we're seeing kind of how we can also generate the alpha ketoglutarate from this, or rather, we can utilize the generation of alpha ketoglutarate by alternatively using this nitrogen on the oxaloacetate molecule. So that's another site that we're seeing. Yeah.
Just follow up. I don't know if you answered this already, but we're transferring to glutamate so we can shunt it into the cell, or is that not one of the reasons?
Exactly. Yeah. So something to keep in mind is that the inner mitochondrial membrane only has so many transporters, and so something you will note, and I would pay attention to what transporters it has and which ones it does not. So a lot of pathways will convert between other molecules simply to get around these transporters, or rather, quite literally get through the transporters. But yes, so no matter the amino acid, you can theoretically have other amino acids, but you want to converge on glutamate. So one, it utilizes the same transporter, and once it's in the mitochondrial matrix, they can all use the same enzyme, rather than having 20 different enzymes and 20 different pathways to get the urea cycle later on. So let's go up a little bit. So again, the glucose alanine cycle, or I skipped a couple slides here. So again, this is the general kind of outline of the chemical logic of these oxidative deamination reaction or transamination reactions, which can, for example, be seen in the mitochondria of a hepatocyte. Now, kind of speaking to the point of glutamine, right? We transport nitrogens in the bloodstream as glutamine predominantly and overwhelmingly so, right? And that glutamine, again, is a more efficient store of nitrogen as you can put 2 nitrogenous groups in it, right? And when I say put 2 groups in glutamine, I mean you're adding 1 to get from alpha ketoglutarate to glutamate. You're adding another 1 to get from glutamate to glutamine, right? And once you have a molecule of glutamate, which can occur via this transamination reaction, we would have to kind of energize it with glutamine synthetase, and that involves a phosphorylation of this variable R side chain. And once it is phosphorylated, you can use the energy to attach on your last nitrogenous group, and now you're storing 2 nitrogens in 1 molecule of glutamine that can then be transported to the hepatocyte for further processing, rather than, again, just producing 1 molecule of glutamate every time you need to get rid of a nitrogen. So the other source is alanine, and other source of nitrogen, right? The other source of nitrogen is alanine. Again, we're thinking glutamine broadly, but alanine specifically in the case of our overworked muscle cells, right? When our muscle cells are overworked, there will be a buildup of pyruvate, and we need to get rid of that pyruvate. One way, we talked about getting rid of the pyruvate before, is by the synthesis of lactic acid or lactate. We spoke about that primarily within the context of getting rid of your NADHs, right? As you have a buildup of NADH in anaerobic respiration, and you need to replenish your supply of NAD+. Now, if we look at the buildup of pyruvate as kind of the central issue, you have one exit via the synthesis of lactate. You have another exit via the synthesis of alanine. So you can attach the nitrogenous group onto pyruvate to generate alanine, and that alanine can then be used for nitrogen transport, specifically from your worked muscle cells, skeletal muscle specifically. And this is known as the glucose-alanine cycle. And when you oxidize your alanine via the oxidative aminotransferase, you will generate you'll generate cytosolic pyruvate. That cytosolic pyruvate will then be used for gluconeogenesis, right? So you can generate glucose from this pyruvate molecule in the same way that if you were to have lactic acid enter that hepatocyte, you would also use that for gluconeogenesis. So now back to the three different entry points, right? We have alpha-ketoglutarate, we have the alpha-keto acids, specifically kind of delineating the movement of alanine, and we're going to generate a molecule of glutamate. But glutamate, again, will be transported into the mitochondria. Glutamine can also be transported into the mitochondria. So, we will want to converge on the point of glutamate, right? So no matter what the amino acid originates from, we want to converge ultimately at glutamine. So that means that glutamine has to undergo an additional deamination reaction. That removal of nitrogen from glutamine will generate glutamate, but that nitrogen removal itself can be used to synthesize carbamyl phosphate. Now, that glutamate molecule itself also, remember, our whole point of generating these molecules in the first place is to get rid of the nitrogen, right? And glutamate still has a nitrogen, so we still need to get rid of it. Glutamate's nitrogens can have two fates. Either they are used in carbamyl phosphate synthase to make carbamyl phosphate. Alternatively, they can also be used to generate a molecule of aspartate from oxaloacetate. So again, another TCA cycle intermediate here that we're seeing is useful within the urea cycle. And remember that carbamyl phosphate synthase is the most important enzyme in the urea cycle, period, right? As it is that rate-limiting enzyme. We will also talk about an additional enzyme that is also important. Sure. But it is that first enzyme that actually kicks off the urea cycle. Everything else can broadly be described as like amino acid catabolism, but this is the first committed step of the urea cycle. So, to make that carbamyl phosphate molecule, what you will do, again, carbamyl phosphate, very kind of strange name. All you're thinking is you're attaching a nitrogen to an energized amino group, and the carbon is kind of what's holding these two things together. Specifically, that carbon comes from the bicarbonate molecule. So how does this actually happen? You have the bicarbonate molecule being phosphorylated. That phosphorylated bicarbonate molecule will then use that energy to facilitate or allow for a nucleophilic attack from the nitrogen. And that nitrogen will then combine with the bicarbonate molecule to become carbamate. And now we energize once more on the other side of the carbamate to generate carbamyl phosphate. Again, that phosphate is not there for no reason. We will eventually cleave it off when we have another carbon adjoining step in our subsequent urea cycle steps. Any questions so far? Okay, so we have our, let's just jump over here. So we have our carbamyl phosphate. Don't forget our aspartate molecule, which will come back into the picture a little bit later. But we have our carbamyl phosphate. That carbamyl phosphate will combine with a different amino acid that we have not previously talked about, known as ornithine. Ornithine and carbamyl phosphate combine to make citrulline. Citrulline will then be energized, and rather than attaching a phosphate group, the way that we energize citrulline is by attaching the AMP molecule. Not dissimilar from the way that we are charging amino acids in the aminoacyl transferase in the process of tRNA synthesis, right? Or tRNA preparation for your protein translations, right? So if you're seeing in both steps, you're energizing an amino acid. In this case, you're energizing citrulline. In the case of the aminoacyl transferase, you are energizing any which given amino acid by making that amino acid AMP intermediate. And both cases, you are, again, making an AMP intermediate. So two cases where you're energizing an amino acid. In both cases, you're attaching on the AMP molecule. So these steps aren't so, they're quite redundant if you understand like the chemical logic behind all of these steps. You'll start to notice that it's not many different steps. Just kind of the same chemical logic applies to different arrangements of carbons and nitrogen. So once you generate that citrulyl AMP intermediate, don't lose track of the overall purpose of the urea cycle. Remember, we want to make a molecule of urea which has two nitrogens on either side of the bicarbonate, or what was the bicarbonate, right? So we're pretty darn close in the sense that we have a nitrogen, the bicarbonate, and our energy that can be left to add on that last nitrogen. And so we expend that energy in the generation of arginosuccinate. This leaves, or rather this uses, a molecule of aspartate which was generated previously in the citric acid cycle, or rather in the urea cycle. Sorry, it is in the overlap between amino acid catabolism and the TCA cycle, right? The urea cycle kicks off with carbamyl phosphate synthase 1, and the addition of the nitrogen to your oxaloacetate molecule exists before that step. So again, the synthesis of aspartate exists within amino acid catabolism, but before that initial step of carbamyl phosphate synthase 1. So now you've left your AMP molecule, you have the arginosuccinate molecule, and so now you're pretty much just kind of like chipping away at a block of marble. You want to get the nitrogens out of there, right? So one way that this is accomplished is you break off one half of the carbon skeletons on one side of your nitrogens. You're generating a molecule of fumarate. Now, once that molecule of fumarate is generated, you are left with a molecule of arginine, which should not be foreign to you, as we talked about arginine in amino acids, right, in general, right? So we're seeing a lot of amino acids, we're seeing a lot of TCA cycle intermediates here, and these structures should not be foreign to you. I will say, I'm not gonna ask you to draw structures on the test, but I would say a good way to like self-evaluate how much you know this material is like, can you blank out all of the intermediates and just like fill it in yourself, right? And if you can draw those carbon structures, you're like definitely good to go, but I'm not gonna ask you to do that. It can be a way that you study and a way to get these things like discerned in your head, but if you don't need to do that, you don't have to do it, but I think it would be very helpful. So you have this arginine molecule, and now you can finally hydrolyze your urea right off of it. And once you do that, you will be left with a molecule of ornithine, which can then be shuttled back. Any questions about this general urea cycle process? No. So, this is just breaking down, again, the chemical logic of the synthesis of the sucralose AMP intermediate. We don't need to dive into that too much. But this is where things get a lot more complicated, right? As we see, there are numerous overlaps between the urea cycle and the citric acid cycle. So I would pay attention to this particular slide, right? As it outlines a lot of potential places where I can ask questions, right? So that aspartate molecule can of course be used to synthesize arganosuccinate, right? The aspartate molecule can alternatively be used as a source of reduction, so it can be oxidized, reducing your molecule of alpha-ketoglutarate and regenerating your oxaloacetate molecule. And also that involves the transamination reaction, right? So once you regenerate that oxaloacetate molecule, you can reduce that and regenerate malate. That malate can be shuttled back into the mitochondria. So if I would probably focus on the shuttles, right? What molecules can actually be moved in and out of the mitochondria. In this diagram alone, we're seeing aspartate, we're seeing malate, we're seeing citrulline, and we're seeing ornithine. Right? And once we start from there, we start to see the different fates of the aspartate. Either it is used to transport electrons when it is reduced to form malate, or alternatively, it could be used to generate arganosuccinate, at which point we cleave off the carbon backbone and regenerate cytosolic fumarate. So we're not just seeing fumarate mitochondrial concentrations, we're also seeing cytosolic concentrations here. That fumarate can then be converted back into malate and then shunted back into the mitochondria. So we're seeing ultimately two different fates of our aspartate, but both of them end with the conversion back into malate. So this is otherwise known as the aspartate malate shuttle. But we will actually get more into that shortly. But before we get more into that, let's talk about the actual regulation of the urea cycle. So again, I would prioritize this as far as information goes, important slide. So carbamyl phosphate synthase, right? This is a rate limiting committing step to your urea cycle. Now, carbamyl phosphate synthase itself, or carbamyl phosphate synthase 1 specifically, itself is regulated allosterically by N-acetylglutamate. Right, so it's regulated by a different molecule. That molecule, the formation of that molecule itself, is in and of itself a regulated process. Right? So how is this N-acetylglutamate actually regulated? Well, let's even take a step back. N-acetylglutamate, all you're thinking, glutamate molecule with an acetyl group attached onto the nitrogen. Right? N-acetylglutamate literally just means acetyl group plus a glutamate molecule. So how do you get these two molecules close to each other? Well, you need the glutamate backbone, you need the acetyl group, and you need some energy to actually facilitate that link. So it's acetyl-CoA. Literally acetyl-CoA plus glutamate. You combine those two together with N-acetylglutamate synthase. Not so complicated, right? Very, very simple. You have N-acetylglutamate synthase. N-acetylglutamate synthase is also regulated by arginine, which makes sense because arginine is featured in your urea cycle, right? So if you have an excess of arginine buildup, you will synthesize more N-acetylglutamate. If you have a buildup of arginine, it's highly likely you have a buildup of glutamate as well, right? Because arginine is downstream of glutamate within the urea cycle, right? Remember, we start out with glutamate even before the urea cycle really begins. So our accumulation of arginine says, okay, we need a green light, and that green light says, let's synthesize N-acetylglutamate. That N-acetylglutamate is in itself a green light to carbamyl phosphate synthase, which says we need to get rid of all these other nitrogens. Right? So several layers of regulation here, and you should be familiar with all of them for this enzyme. Right? So again, from the top, accumulation of arginine, likely from the buildup with the urea cycle, will say, let's use our excess glutamate, which again is implicated in the urea cycle, and let's use our excess acetyl-CoA to generate N-acetylglutamate. The N-acetylglutamate says, okay, now let's get rid of our other nitrogens, right? And hopefully you're getting rid of other arginines, you're getting rid of other glutamates, so that this regulation in and of itself can provide that negative feedback loop. So you're actually limiting your accumulation. It's not just going on forever and ever and ever. So this is that graphical explanation, right? So we have acetyl-CoA plus the glutamate generates N-acetylglutamate, which acts as a green light. So this means kind of propels in the forward direction for carbamyl phosphate synthetase 1. Now, that synthesis itself is also regulated by arginine. So these molecules are implicated themselves in the urea cycle. So pay attention to those overlaps. Any questions on this kind of like multi-level of regulation here? I know it can get a bit confusing as it's like quite redundant, but it's all important, I would say. Yeah.
So it's like, it's activated in general by like high levels of nitrogen, because if it's activated by the presence of ammonia, but also the presence of glutamate, is it going towards putting the nitrogen into like storage stuff, or is it going from urea to storing it, or from storing it to, like, I'm confused what it is.
Yeah, so urea ultimately is the end product here, right, that you're going to dispose, right? So if you're generating urea, I don't know of a pathway that uses urea as a substrate to make other things, right? So you're going to use this urea, it will be excreted, and so on and so forth, right? So you're not thinking you're going to recycle this urea, but you're trying to get rid of these excess nitrogens. And two signs that you have excess nitrogens, right, is increased arginine, increased glutamate, right, which can both be used to accelerate the disposal of the ammonia itself. Yeah, does that make sense? Any, yeah.
How does acetyl-CoA relate? Why do you need high levels of that?
Acetyl-CoA is used for so many different things. It's hard for me to kind of like lock down a particular one, but it's just generally present in high concentration. Pretty much anytime you need a carbon skeleton, acetyl-CoA will kind of come to the rescue. And again, think of it as like carbon backbone being held by the CoA molecule. But it's an easily removed carbon backbone because of that thioester linkage. Any other questions about this slide? Again, I understand it can be a bit confusing and a bit iterative. So if it needs a second or you want to save it to Wednesday, that totally makes sense. So again, we're thinking we're thinking also this is happening within the liver, again, right? You would not expect your arm to start producing pee and then all of a sudden you have to like get rid of it from your arm, right? It's going to be heavily localized. There's like actual transport. Yeah.
Is my other question is from carbamyl phosphate, how does it get to urea?
From carbamyl phosphate?
Yeah.
So then it will combine with the ornithine molecule and then citrulline is transported.
So that goes directly to step 1 of the urea cycle.
That is step 1 of the urea cycle. It is the I think 1 is just denoting what is on the slides here, but the first step of the urea cycle is the synthesis of carbamyl phosphate.
It looks like it's going from it and then phosphorylating it as the first step.
I think it's a matter of like how the slide actually describes the numbers, but it's widely considered that carbon, like here it says 1, it says 1 there. I would definitely consider carbamyl phosphate synthase as the first step to committing to the urea cycle. Back.
In the one where it shows combination of citric acid cycle, urea cycle, and the other one, there's aspartate is coming out of mitochondria, and then it goes one of it in argininosuccinate, and the other one is transported transport to oxaloacetate, does it mean that some of the aspartate goes into one way and other parts of aspartate go into argininosuccinate circle?
No, it's saying that there's two different fates for the aspartate. Either it goes into the argininosuccinate pathway, or alternatively it goes into the oxaloacetate.
So some goes this way, some goes this way.
Yeah.
Okay.
And we'll have a more detailed slide on it. Okay, so we don't need to talk about all the different pathways for amino acid oxidation. As you can see, broadly, a lot of different pathways here. And they all use a different network of enzymes, which also overlap with a lot of the enzymes that we talked about earlier. But I broadly kind of skip over these as it's kind of just rote memorization, and you don't need to go over that. Did someone have a question? Yeah.
Just to be clear, carbamyl to carbonyl phosphate, that's where the synthase is involved in, right?
Carbamyl phosphate synthase it first facilitates the addition of the phosphate group onto the bicarbonate, and then the addition of the nitrogen to replace that initial phosphate group, then the addition of another phosphate group, and then that is the complete reaction.
So where do you see the rate limiting substance, the rate limiting enzyme?
Where do you see what?
Yeah, where do we see the carbonyl phosphate synthase one? At which step?
That is what kicks off the urea cycle, and the carbamyl phosphate synthase, or the carbamyl phosphate, is then used to create.
Which reagent and which product? Where do
You
see
use
it?
bicarbonate, you use 2 molecules of ATP, and you use your nitrogen.
So this whole process is using that one enzyme.
Mm-hmm.
Okay.
Yeah.
This is crazy.
Any other questions about? Yeah.
Wait, so is the generation of citrulline step 2 then? Or I guess-
Yes, I would consider the generation of citrulline to be step 2. Yeah. So just think, what kicks off the urea cycle, carbamyl phosphate synthase 1, and the regulation of carbamyl phosphate synthase 1. So that's N-acetylglutamate synthase. That's the allosteric regulators of that, so arginine, right, and N-acetylglutamate, which allosterically regulates carbamyl phosphate synthase. Beyond that, I'm not going to be super nitpicky on like this is one step of the urea cycle, this is another step of the urea cycle. I think where this is difficult and where I would expect to ask questions is in the overlap between the pathways. You would expect an increase in this, or what would happen if you had this kind of cell in this kind of condition? How would that affect these different pathways? Right, so I think it's difficult enough with the different shuttles. I'm not gonna berate you on like, oh, this is step 1, this is step 2, this is step 3, this is step 4, or memorize every single enzyme. I think it's a little bit too much. But that's not to say it won't get more confusing, because it will. So, we kind of started off with this initial kind of introduction to the malate aspartate shuttle. But it gets more nitty-gritty than that, and this part I do think is important. So, the malate aspartate shuttle is actually 2 antiporters. What is an antiporter? An antiporter transports 2 molecules going opposite from one another, right? So, the malate aspartate, malate part is actually a malate alpha-ketoglutarate antiporter, and the glutamate part, or the aspartate part, is actually a glutamate aspartate antiporter. So get ready for things to get more confusing. So we have our aspartate molecule, right? The aspartate molecule will be shuttled out of the mitochondria. With it, in the opposite direction, it will be accompanied by a glutamate molecule. Right? So that glutamate molecule could theoretically enter those initial steps of your amino acid catabolism. So one piece of overlap between 2 different pathways. You have the aspartate and you have the glutamate. The glutamate is entering in the amino acid catabolism. The aspartate is leaving in the urea cycle. Now, that glutamate molecule itself can be generated by mitochondrial alpha-ketoglutarate. that again occurs via this transamination reaction. Right? So that alpha-ketoglutarate will undergo a transamination reaction, generate the glutamate, be shuttled into the mitochondria, where it can then undergo these deamination reactions. Right? But that alpha-ketoglutarate can get there in the first place via the malate alpha-ketoglutarate antiporter. Right? Moving alpha-Kg out of the mitochondria and into the cytosol. So, with it, it will bring malate in the opposite direction. So we now know of two different concentrations of malate, cytosolic malate and mitochondrial malate. So when we generate that molecule of malate, or we transport that molecule of malate back into the mitochondria, we can oxidize it once more to generate oxaloacetate, and that oxaloacetate, as we know, can be used to generate our aspartate once more, kind of closing the loop here. Now, we can also consider why we are doing this in the first place, right? The purpose of that inner loop, right? The inner loop of oxaloacetate to malate and the reoxidation of malate to oxaloacetate. So previously we talked about how NADH is free floating, and we also spoke about the oxidation of NADH in complex 1, right? Our complex 1, while the NADH docks at complex 1 and NADH is mobile, the movement of NADH across the inner mitochondrial membrane itself is impossible. NADH cannot freely traverse the inner mitochondrial membrane. It can freely move throughout the cell, but specifically it cannot penetrate the inner mitochondrial membrane. Remember, the inner mitochondrial membrane has two characteristics which make it particularly impermeable. One is the presence of cardiolipins, which has those 4 fatty acid chains attached to it. The other is the high lipid to protein ratio, or high protein to lipid ratio, meaning there's a lot of proteins to very few lipids. And it's much harder to navigate through an enormous protein complex than it is to weave between a couple of fatty acids. So, what we do is we use oxaloacetate. It's essentially a temporary storer of these electrons. And we reduce our oxaloacetate and we reoxidize it on the other side to regenerate that oxaloacetate. And when we reduce it, we generate malate. So, we are introducing these two antiporters within the context of the malate aspartate shuttle. This links together the TCA cycle, links together your urea cycle. Additionally, this loops in the electron transport chain, as we introduce the problem that we can't actually move the NADH across the inner mitochondrial membrane. So you're linking up essentially 3 different discrete units here with this one slide. Now, we add on the additional layer that this is happening predominantly in the liver, kidneys, and heart, right? So other muscles which don't have as high energy requirements or have different energy requirements than liver, kidneys, and heart will have to do a different thing, right? This is the glycerol 3-phosphate shuttle. So this happens in skeletal muscle and the brain. So again, this is more so kind of addressing, okay, so we understand that NADH cannot penetrate the inner mitochondrial membrane. So what happens in things that are not our previous slide, right? What happens in the other cases? In the case of the skeletal muscle and brain, which have excess levels of glycolysis, we can use our dihydroxyacetone phosphate as a very temporary storer of these electrons again. So previously we saw it with malate. Now we're seeing DHAP being converted to glycerol 3-phosphate. Glycerol 3-phosphate is not GAP. GAP, glyceraldehyde 3-phosphate, or G3P, glyceraldehyde 3-phosphate. 2 different abbreviations for glyceraldehyde 3-phosphate. Meaning, same exact structure, except it's an aldehyde. Here, this is glycerol 3-phosphate. Remember, 3 carbons, hydroxides, and also the phosphate group on the side. Glycerol 3-phosphate can be oxidized, generating a molecule of FADH2. Now, this FADH2 will be oxidized to generate ubiquinol, The specific enzyme that facilitates this is mitochondrial glycerol 3-phosphate dehydrogenase. Don't need to get too into the weeds with regards to that specific enzyme, but it is important to pay attention to the fact that this ubiquinol goes directly into complex 3, right? So you're missing out on ATP that can be generated from complex 1. More or less, that's fine for these specific tissues. So you are missing out on ATP that can be generated from complex 1 via that pump in complex 1. That is not happening here. But that's fine for these particular tissues and muscles. We can continue on with complex 3 and be just fine. You're still doing oxidative phosphorylation, but the amount of ATP you are generating per complex is now reduced. So again, recap, we talked about the malate aspartate shuttle. We dove a little bit deeper, talking about the link between the TCA cycle and the urea cycle and the electron transport chain. And then now here, we talked about a specific overlap between the electron transport chain and actually glycolysis in specifically the skeletal muscle and brain to address the same thing that this does for our liver, kidneys, and heart, right? Which is, again, the movement of electrons. So I iterate, in skeletal muscle and brain, you will not see these antiporters being used. Okay, any questions about that? Okay. So, now we'll do like a very quick debrief on like metabolism and epigenetics. We won't talk about it a lot. Okay, how many of you guys have learned about epigenetics previously in a bio course? Okay, pretty much everyone. So this shouldn't be like too complicated. How many of you guys have heard about like methylation specifically before? And acetylation? Okay, so you guys have covered, I think, the basics. Can someone tell me what they know about acetylation and methylation?
Acetylation promotes transcription while methylation does not.
Yeah, right. So I totally agree that that's what they taught me. But as we know, these things get more complicated. So I will say acetylation. Well, okay, so we think about the interaction between a histone and a DNA, or like a strand of DNA. Broadly, right, we can think that we can modify the availability of the DNA and stop transcription from even occurring in the first place. We are seeing that now it's not just a one-directional thing where modifications in the DNA can lead to translation of specific enzymes, which lead to the accumulation of metabolites. It's not just a one-way street. What we are also noticing is the metabolites can go back to the nucleus. Some of the enzymes that are actually translated can go back to the nucleus and modify the DNA and histone proteins themselves, now kind of closing this loop, right? So it's not just nucleus tells what metabolites to create by one way. We're now seeing those metabolites can go back, close the loop, and influence the nucleus. So acetylation, right, takes advantage, or rather, any of these modifications take advantage of the fact that our DNA is negatively charged, and our histones contain, most often the residue that is talked about are lysines. Lysines which are positively charged. So we take advantage of the positively charged lysine and the negatively charged DNA, and those two things, by virtue of being oppositely charged, can interact strongly with one another. So when they do interact strongly with each other, we have something known as we have our tightly bound DNA across our histone proteins, and this is known as heterochromatin, right? Now, we can undo one of those charges. We can undo and get rid of the positive charge on the lysine residue by attaching an acetyl group onto it. And again, we're talking specifically about the lysine residue of the histone. Now, we can also methylate. So this is kind of broadly just showing. So we're not going to talk super in depth about all of these different epigenetic or the different enzymes involved here, but we will focus on a couple of them. So our acetylation can undo the opposite charges and allow for the DNA to unravel, generating U-chromatin. This U-chromatin can be freely transcripted, or you can generate transcripts from this U-chromatin, and that will go into translation. Easy, right? Methylation, on the other hand, essentially locks these things in place. And methylation will prevent our DNA from unraveling from these histones. Acetylation, universally affiliated with transcription, translation. Methylation is more complicated, and it's more broadly established that it can do either. It depends which site you're methylating, and it can either release that DNA from the chromatin or tie it up in heterochromatin. So methylation, mix, can do either or. But acetylation pretty much universally just unravels that DNA. Methylation a little bit more complicated. Now, with any epigenetic modification, and we're not just limited to those on the slide, we have the addition of succinate, we have the addition of fumarate, we also have the addition of lactate, but other things, other metabolites can be added. But in any case, with epigenetic modifications, they can either be attached or removed, very simply, right? In the case of lactylation, they still don't know a lot of the enzymes that facilitate this, but in the case of demethylation or acetylation, very well studied. Methylation, less well studied, and other modifications, significantly less well studied. So we can think acetylation is facilitated by histone acetyltransferases. These histone acetyltransferases will use acetyl-CoA as that molecule to add in that acetyl group. Pretty simple. Now, there's a class of enzymes known as sirtuins. Sirtuins will remove the acetyl groups. We won't talk too much about sirtuins. But the generation of acetyl-CoA here is something to pay attention to. And it should be noted that this is nuclear acetyl-CoA, right? So nuclear concentrations of acetyl-CoA. Now, if you look at histone methylation, facilitated by histone methyltransferases, simple. Removed by histone demethylases, simple. Classes of enzymes gets a lot more complicated than that, but we can just consider it in the classes of demethylases. Now, histone demethylases utilize alpha-ketoglutarate as their substrate and generate a molecule of succinate. Again, this is all nuclear, so you need to have nuclear alpha-ketoglutarate here. So we will focus in on the acetylation and the demethylation, and we will talk about two specific overlaps here. There are many other such overlaps that exist. So histone acetyltransferases. Here you're just seeing a specific residue that has the addition of the acetyl group, and you're literally just attaching it on. So not particularly complicated there. So the acetylation itself uses nuclear acetyl-CoA lyase, right? So, or nuclear ACL1. So where else have we seen ACL1? Where else have we seen this enzyme? It's in the asynchronous lecture. That maybe most of you have not watched yet. Okay, it's in lipid metabolism. What was that? ACL1.
What is it called?
Acetyl-CoA lyase. Oh, I didn't hear. Sorry. But you have the generation of your acetyl-CoA molecule, and you are cleaving it off from your citrate molecule, right? Think citrate, think 6 carbons, right? And your CoA. So you cleave it off, and you're generating via acetyl-CoA lyase, you're cleaving off that acetyl-CoA, which can then be used directly in your histone acetylation, right? But this is now establishing the relationship between these mitochondrial transporters and actually what can happen in the nucleus, right? So anything that upregulates citrate within the mitochondria can be shuttled out and then shuttled back into the nucleus, so out of the mitochondria, into the nucleus, and used for your histone acetylation. Again, unraveling the histones. Yeah.
Oh, wait, so it turns into oxaloacetate and it's turned into malate, or?
Yeah, it's turned to oxaloacetate. The oxaloacetate can be used, I don't know all the pathways that oxaloacetate can be used in the nucleus. Yeah.
Because citrate's in a cycle, like what would cause it to build up, I guess?
Anything that would like cause you to upregulate the TCA cycle, right? So maybe anything that would cause you to upregulate the TCA cycle. So if, for example, you're exposing to oxygen, right, then all of a sudden citrate synthase will start working in overdrive. You have more citrate temporarily. That citrate can be shunted into other pathways, but also upregulate the TCA cycle in and of itself. Yeah.
So then, does that mean if the TCA cycle is upregulated, then there's more acetylation in the nucleus in general, or?
No, I wouldn't go that far, simply because I don't know the experiments that have like established that. I'm careful to like overpromise here in terms of what has been established in terms of information. All we know is that citrate can be transported out of the mitochondria into the nucleus and used for histone acetylation. Yeah.
If like you needed to upregulate nuclear citrate, would you upregulate like the TCA cycle? Can you more citrate to acetylate?
Yeah, sure. Yeah, your TCA cycle citrate is a citrate that is used ultimately for your nuclear metabolism.
Right.
There also remains the hypothesis, like, is citrate generated in the nucleus? To which I don't know, and I don't think the research community. But it is a very interesting area of research, is like what metabolites can be generated in the nucleus. Therefore, kind of, you know, what we're seeing is a lot of the enzymes in glycolysis can be moonlighting. That's what they call it, is that they simply take on an additional function within the nucleus, so they can actually translocate. And it's the same protein. Like, it's like it will move from glycolysis and move into the nucleus. So the canonical activity of these enzymes is largely unknown.
The main source of like citrate is the TCA cycle, right? There are
Yes,
like other.
as far as now is concerned, yes. Citrate coming from the TCA cycle. Yeah. Any other questions about this slide? Okay. So now let's very quickly cover our demethylation, right? So we talked about histone acetylation. Right now we're going to talk about demethylation. So I did say demethylation or methylation is kind of mixed. Like, does it facilitate translation or transcription or not? I will say oversimplification is we can say, for the intents and purposes within this class, that it does facilitate so acetylation wraps it up, methylation we can think will kind of unwind it. Right, or sorry, acetylation will bind it up, methylation will do the opposite. Right? Again, acetylation will neutralize your positive charge, right, allowing for the generation of euchromatin. Methylation will increase the affinity of the interaction between the histone and your DNA complex. It is mixed, but within the context of this specific reaction, it is not mixed with these specific methylation sites that we are mentioning. And generally, when you see methylation sites, it's just are like histone modifications. This means histone 3, glycine 9, and you're adding 3 methyl groups onto it. And you can add multiple methyl groups onto a particular lysine residue. But as we had mentioned before, demethylases use alpha-ketoglutarate as a substrate, right? Generating succinate in the nucleus. Now, we can have molecules that look a lot like alpha-ketoglutarate interacting with the same enzymes that alpha-ketoglutarate does, therefore acting as an inhibitor. So one inhibitor, or one molecule, which has 2 chiral forms, is 2-hydroxyglutarate. 2-hydroxyglutarate can be generated in 2 different ways. Or rather, it can exist in 2 different chiral forms, which themselves can be generated in different ways. One chiral form is via lactate dehydrogenase A and malate dehydrogenase 1 and 2. Now, the conversion of alpha ketoglutarate to L